can we create a simple and effective solution for bone density loss in space?
As for the current state of this problem, there are some things being implemented, such as bisphosphonates and gold nanoparticles, which are being researched into. But these aren't the most effective solutions, so that's why we aspire to create a more effective solution. Adi, would you like to explain a bit more about the available solutions?
Sure. Bisphosphonates are medicines that have been proven to prevent bone density loss. They've been used in the past to treat osteoporosis, and people thought that if it can treat that, it'll be able to treat the bone density loss that astronauts experience in space. An example of a bisphosphonate is alendronic acid.
Now, about our solution, we decided that rather than focusing around the problem symptoms, we should focus on the problem source, as in tackle where the bone density loss is occurring. So for this, we decided that we should start off by trying to change the DNA of the astronauts using a gene editing tool called CRISPR. This would allow them to produce more bone density in space while maintaining normal levels or on Earth to try and balance it out and keeping the bone density at normal. So now we'd like to start off with a small interview of you. So before we start off with the formal part, could you please tell us your profession, like what you studied in university? Would you mind saying that?
Sure. So I have a master's in genetics, a PhD in molecular and cellular biology, and a postdoc in immunology. And I'm currently a sessional lecturer at McMaster University.
And I have a master's in geology and a PhD in planetary geology. And I'm currently entrepreneur.
Okay.
And educator.
So, we had a few questions for you and specifically.
Sure.
These are questions on the medical side. Adi, would you like to start off with the first one?
Yeah. Why do osteoblasts slow down their activity at microgravity while osteoclasts continue to work at regularly?
So that's something that we don't actually fully understand. Why is it that the osteoblasts versus the osteoclasts actually have a different development rate compared to what's happening on Earth? There's a lot of research happening to try and understand that, and they're doing the research in mice mostly. It's got to do with the actual development of the cell. So the osteoblast is what creates the osteoclasts, right? They will differentiate to develop the osteoclasts themselves. There really isn't any understanding why does the proliferation decrease. We have no idea.
Okay. So, do any of the current solutions to bone density loss have any side effects?
Do any of the solutions have any side effects? You mean any solutions to bone density loss?
Yeah, like bisphosphonates or like anything used to treat either osteoporosis or bone density loss in space.
I don't know.
Are there any on Earth? I mean, as in to a patient who may get a certain treatment?
Not that I know of. It's something that I can look into and get back to you, but I don't think there's any detrimental side effects that we know of. Yeah, I can't think of anything. But I will look into it.
Okay. So, what genes are involved in the process of bone density loss, and how are they involved in this?
Oh my god. I know that I sent an email about there was a particular protein that they found. So my expertise is not in bone density. Just to make that clear, I study the intestines.
Oh.
So guys asking some very specific questions. I can pull out the email that I had sent that actually talked about the specific protein that they found in mice, at least, that was being decreased in expression and might be related to bone density itself and for loss in muscle mass. That is not proven. that is not a definitive answer. It is just one protein that might be associated with it. So it's not fully understood. Like what you guys want to do is great, but there isn't one particular gene or there isn't one particular protein that's been narrowed down to that this is exactly the target that we need to be working at. There is a lot of research still required to understand how gene expression changes in microgravity, why it changes in microgravity, and what proteins are actually associated with the different losses that do happen, like the changes that astronauts see. Yeah. So if you're looking for like a specific name of a gene, there isn't one yet. But
And do we know like how many are affected by it?
that is some work that is being done. So if you remember when, I think it was earlier this year, it broke out into the news that, when they compared an astronaut and his twin, and they talked about how the genes had been changed in the astronaut. That's not exactly what happened. The genes haven't been changed. It was slight changes in expression, but there's a lot of research that still requires an understanding what are the particular changes that take in place, if there are specific genes, or is it just overall ups and downs, and whether any of those actually have a significant value. We don't know. So that's not something that's understood yet.
Right, Adi, would you like to ask the next question?
Sure. What is your opinion on our bone density balancer in way of solution?
No,
Oh,
this
sorry.
one.
What other health problems do you think astronauts face in space?
You might have.
I
a
think
better answer.
I had mentioned a few
Yay.
in terms of bone density and then like heart rate and like getting the blood flowing through the system when you're in a microgravity.
Yeah, that's a big one.
And then, I mean, the solution is just you having to be active and having certain hours spent in your work schedule to be physically moving, just so that you can not only slow down bone density loss, but at the same time ensure everything is moving and,
Because that affects, not only is it blood flow, it affects nutrient uptake, it affects how oxygen is actually being passed to all of your cells in the body. It goes down to your brain function as well. So that's a big one that astronauts have to deal with. There's also in terms of eating.
Eating and like dilating of eyes. I don't know what the.
Really?
Well, I mean that's a concern. Like, because again, in microgravity, your body's sort of loosening up, right?
Yeah.
And also bones start to grow longer. You incrementally grow taller. Right, like because it's all relaxing, so gravity is not holding it together. So those are, I mean, side effects that start to really manifest once you're back on Earth, because now everything is trying to figure out how to get back to its original self after going through this. So that's one challenge.
You said you studied intestines, right? So can microgravity affect that?
I think so, because at the end of the day, when you're talking about digestion, you're talking about nutrient uptake. In the lack of microgravity, you're not going to have proper digestion that's going to take place, because food does move down your esophagus, it moves down like past your intestine, but that's actual muscle movement that does it, that's not gravity that's doing it. So it'll still go down, but having said that, you're also losing muscle mass. So how well your intestines can actually help with the peristalsis it's difficult. So in terms of, the actual nutrient uptake, there is an issue there. The other thing that I can think of, I just had it. Food, intestines, food. Oh, the microbiota that's in your intestines. That starts to become another concern, because when you are in gravity and your cells are also starting to kind of loosen, it's the adhesion between them that starts to loosen up as well. So that can cause whatever bacteria you have inside your intestines to get a little bit of a leeway to enter your body. So infection can also be an issue that way, because now you're getting bacteria that's supposed to be outside your body going inside your body. because they don't sterilize their stomachs when they go. Like astronauts don't do that. So, and then I don't know if there's any research being done on what the microbiota looks like in space compared to what it looks like for a healthy individual on Earth. I don't think that's even been touched into yet.
Or publicly released as info.
Yeah.
Or data.
We also have another question. This is more of an opinion question.
Mm-hmm.
Do you believe there is a multiverse?
Yes. Don't ask me why. I just do.
That would be part 2 of it.
Okay, so having studied evolution, having studied how humans have come on Earth, Earth is not the only planet that evolution could have happened and intellectual beings can survive. So in terms of like multiverse, I do think that there's somewhere else in this vast universe of ours that we have another somewhere. But you are the one that's more...
I think for this question I said I didn't know enough about multiverses to give a sort of answer.
Fair enough.
Because, yeah, it's like multiverse would be several universes. And that it leads to me where I was like, oh, I don't know enough about that. But in terms of finding other Earth, like, planets and things within our own universe, sure, that is a very common thing. If anything, it's the challenge is how we find Jupiter-like planets so close to their host sun that is almost like where Mercury is to our sun. So that's its own sort of how did that happen? But and that's sort of a different approach to what is a multiverse? Right? So,
I guess I didn't think of it that way.
So, as we talked about our solution earlier, we also had a name for it. We called it the bone density balancer. So, do you have any like feedback or opinions on our solution?
Right off the bat, when you talk about your solution about changing the DNA of astronauts, I think that's a bad idea when you put it that way. Because the DNA of astronauts is not what's changing when they go into space. It's not the DNA, it's the expression of the DNA. It's DNA being expressed into RNA and then making protein. DNA stays the same no matter what. So when you say you want to change the DNA of the astronauts, you are suggesting that you need to quote unquote mutate these individuals to be able to go into space. Not that like I believe that mutations are bad or anything, but I don't think that is a valid solution because you need to be able to control the expression of the DNA that you're also introducing. And so if you don't understand how the expression is changing in going into a microgravity environment versus what's happening on space, it doesn't matter whether you add more DNA into the individual. So I think like the name in terms of like it being a balancer is good, but I think where that balancer comes into is how do you work on the expression of the genes and balancing the expression of the genes rather than trying to add more DNA.
So for that, we also said that like the gene could be like we were imagining as if the gene could be controlled based on the microgravity, right? So then it could be expressed in a higher during microgravity, whereas it wouldn't be expressed during higher amounts of gravity. So then, would that be suitable for it, or like?
Yes and no. Changing the expression of DNA, yes, that is suitable. How do you change the expression of DNA in terms of the level of gravity someone is feeling is the big question. How do you suggest to do that?
Well, your body has an internal sense of gravity in a way, because we can, like your body can actually kind of feel that, your lack of gravity in some cases. So then we were thinking that that might be due to something internally. So if we could somehow find a way to express the gene according to that, then that might be possible. We and you also sent the paper based on the protein, right? So then we believe that that might be a possible way to do that.
So the paper that I sent about the
Yeah.
protein was a specific target that you could be looking at. But
Yeah.
how are you going to control when, because scientists still don't understand what changes the expression in microgravity versus having normal gravity. We don't know what actually causes that change. So how do you propose to use that system to control gene expression? Because yes, there is an internal, the body does know when there is sufficient gravity. That's how we evolved. But when you're saying to use that internal system, what is this internal system that you're talking about?
So we should consider that, yeah. So do you have any idea about its feasibility? This idea?
In terms of using CRISPR,
Yes.
again, using CRISPR is not the most conducive solution, because what you're going to do is add more DNA. And that's not going to change the expression of the DNA. It just adds more DNA into the system. So I don't think that that is the most feasible solution. There was a second paper that I sent that talked about how you can use CRISPR to control gene expression. Did you guys look into that?
Yeah. So is what you're saying is that if we used CRISPR to control the gene expression, that and rather than adding in our own gene, that would be a better solution.
Yes. Using CRISPR to alter gene expression rather than adding DNA. So you change the expression of existing genes.
Okay. So what percentage then, or does
Well,
that
That,
play a factor?
that will.
Yeah.
So that's a question to you guys, is how are you going to control how much expression is normal in this microgravity versus, so how do you tell the body, now you have normal amount of expression and maintain it at this, rather than going over and having too much of it, versus them coming back to Earth and then being like, okay, now you have too much of this expression, turn it off so that we can come back to normal. How do you control that?
So
What you can do is probably you can actually take the current expression while you are on the earth, and when you are actually going into the space, there will be a difference. Then what you can do is you can probably see whether adding additional, as if you are mimicking you are in the earth while you are in the gravity.
So set the Earth
set the-
value as a normal.
Exactly. Then while you are coming down, make sure that is not getting, meaning decreased. If it is getting decreased, then that means pretty much I need to offset the balance so that everything is equal. While you are in the quiet period on the earth, that is where you need to make sure whatever you had in the past before leaving to the space, you maintain that. Probably. If you are able to control the expression, it's like a kind of giving an injection or something like that. I don't know. I'm just throwing an idea.
So
Right.
that's a very mathematical
Yeah.
coding type solution that you guys are giving.
Yeah.
Biologically, to say stop when expression is what would be at Earth is a lot more complicated.
Yeah, maybe it's a conceptual idea. Maybe probably because I know this entire solution, what we are trying to propose, might involve a bigger audience and bigger discussion, etc.
Yes.
But at the same time, from the conceptual presentation perspective, we can always say this is the approach which we're planning to take it.
So I think that second paper that I'd sent that had the CRISPR activation with the CRISPR inhibitor in the same cell, that is a good method
Yeah.
that you can propose.
Okay.
The reason I say that is because the idea behind that is that you can have the CRISPR. So do you guys understand how CRISPR works?
So I'm not really sure like how much we understand as a whole, but I do believe that we understand that CRISPR works as a gene editing tool based on like, so there's a guide RNA and then there's the actual Cas9 protein. So Cas9 is the protein that does the actual cutting and insertion of DNA, whereas the guide RNA is just like a template for the Cas9 protein to follow.
Right. And then,
Everyone follow that?
so the thing is that you have your guide RNA. That guide RNA is what you can change to whichever template you want. And so you can add your gene of interest into whatever your, like wherever it is that you want to add into the genome. With the CRISPR activation and the CRISPR inhibitor is that they're two separate ideas. One, it's still with the Cas9, the cutting, adding, but what you do is, in one case, you add your expression level. So the RNA is attached to something that is in the cell that is going to control your gene of interest. So it's something that will actually normally activate and get the expression of your gene of interest. So you have extra of that, and at the same time, you add another one that has something that will turn off this gene and have that.
It's like a little labor book.
Yeah.
Like a
so
switch
it's-
on and switch off.
Switch
Yeah.
on and switch off.
Okay.
So what happens is if you put both of them into the same cell, because your natural activator turns down when you go into space, you have this extra gene present to try and activate that gene. So you have this greater probability of keeping an activation of the gene. And then when you come back on Earth, because you're getting overexpression of this gene,
Turns it off.
the one that
The
turned it
inhibitor?
off, you'll have that kick in and turn it off. So you can now maintain a balance of how much your gene actually expression is required. So instead of you guys actually talking about we will use CRISPR, I think you need to actually talk about how are you going to use CRISPR for the gene expression and allowing for that balance itself, so that it doesn't sound.
Too scientific.
It's not that it sounded. You want to sound scientific, but you do want to. You don't want to just say we will use CRISPR
Without any
for gene
reasoning
expression.
behind it.
You want to give reasoning, and you want to show that there's a feasibility because you've looked at literature.
Yeah.
It's
So-
not like this is just a concept we came up with.
So would adding an inhibitor or anything change the cost of the solution, the overall cost?
It will.
So how much would it increase or decrease by, approximately?
You could probably double the cost easily.
Just by adding an inhibitor and activator gene.
Yeah, because now you're not just talking about using one CRISPR, you're talking about using two different solutions into one cell.
So we were researching online and we found that the current cost for gene therapy, which is in a way similar by using CRISPR, is around $10,000. So do you think that by doubling the cost, that would mean this would approximately cost about $20,000, roughly?
Yeah.
Adi, would you like to ask the next question?
Do you have any innovations or ideas on your own to solve a similar problem?
So, have you ever come up with any ideas of your own?
I just gave you all my ideas. I looked into it and I was like, oh, wait, hold on. You could do it this way. Do I have any other ideas on bone density?
No, not exactly bone density loss.
But
Just
just
like
the idea of
any
using
problem
this.
that you wanted to solve, did you ever have an idea that what if it could be like this and that would be like a solution for it?
Were you using this activator inhibitor?
No,
Or
just
Just
anything.
in general.
In general,
Like, for example, if you've ever had a problem or like a solution to solve the problem of littering or something, it doesn't have to be on topic, but like, it's just an opinion question.
A problem
Ha-
is a solution.
have you ever approached a problem, and then have you ever thought about how to solve it? That's what he's
That's
asking.
what
That's
we do
kind of what we
every day.
do on a regular basis as
For
scientists.
daily life.
It's actually, I think what they're trying to say is, have you ever thought of a problem, any problem, like just on a broad spectrum? Have you ever thought of a problem and tried to solve it in a similar fashion to what we've done?
And have your innovations ever been put into use or like tested or something?
Oh, so you mean like, have I actually come out with a
Yeah,
product?
yeah, exactly.
Okay, because I was like, I mean, I'm a scientist, I come up with problems all the time, and I try and find solutions to them all the time. Anything that's actually being put into practice. Is it bad that I don't say like, no?
No, I don't think I have any either.
It's, growing up, I didn't have opportunities like you guys do right now to come up with these pretty innovative ideas. There was once we tried to make a robotic car at home. My brother and I. That didn't go too well. My parents were not very happy that we took apart a lot of the electronics at home, and they were. I got a lot of scoldings.
Did I give an answer for this last time?
No.
You
Yes,
said
you
that like I believe you said you did have an answer, but like it was more related to the scientist aspect of it, but not like any product or anything.
Okay, so because now I'm thinking on this, I'm thinking back to grade 11 when we had to,
Did you make the catapult?
not the catapult, this was tech class, so like we had to design a aerodynamic vehicle that had a carbon dioxide cylinder inside as its propellant.
Oh,
We had to design that and then do the test, and clearly I saw it was a finished product. I think I have it somewhere in the house. But mine didn't end up winning amongst all of us, but that was probably one where we had to think about what was it that would make it aerodynamically smooth to go a certain track length, and how did moving that from a design print into an actual, like, we had to start with a piece of wood and then design on it, cut it, and smoothen it out and stuff, and then launch it with those cylinders. And then it was a neat sort of train of thought to go through. So if anything, that for me is a product that ended up being.
I did do that in grade 12 physics. And
Ours was tech class.
we had a race at the end. The whole class would make their own cars, and you started off same thing with a piece of wood.
Yeah.
And you could only, so we didn't have necessarily, it had to be a gas cylinder type of aerodynamic kind of idea, but the idea was you are going to create a car using rubber bands and anything else you can find in a grocery store specifically.
Okay.
And it had to go on its own in a straight line, and everyone who did we just.
So I guess that is like, prototype of a Lego thing, but
Yeah.
we didn't have this,
We didn't
but we clearly had to probably go through a similar thought process of
Yeah.
how would you design it, and some made fascinating vehicles.
I mean, in my research, I created a transgenic mouse that is a model for inflammatory bowel disease,
That is neat.
but that's like kind of my job, so I don't
No,
know.
but
if
it is also being used now.
We're trying to get people to use it as a model for inflammatory bowel disease, yeah. So I did make and characterize this mouse,
There you go.
yes. And mutate mice,
It was a live mouse.
yes.
Oh.
How about you?
Oh, never
Mine.
mind. I thought it was something else. Oh.
But,
I studied mice. I did a lot of my research. So my PhD research was actually creating this transgenic mouse, and then I characterized it and found that it had inflammation in the intestine, and realized that it looks very similar to inflammatory bowel disease in humans. So in humans, we have no idea why inflammatory bowel disease happens. And anything that we want to study in mice, we actually use chemicals to create the inflammation in the mice. Now I have a mouse model where it spontaneously gets the inflammation that is very similar to what happens in humans. So now we can study and understand why inflammatory bowel disease happens and what kind of therapeutics can we use to not only maintain it but to prevent it if we catch it early on. So I did that, and then in my postdoc in immunology, I actually studied why does peanut allergy develop and why is it lifelong for some people. So in mice, I worked on how to make mice allergic and studying what happens in your body when allergy takes place. And the reason is that in humans, we have no idea how or why allergy happens, and it only happens in some. But especially with peanut allergy, it's for 80% of people, it's when they're babies, and for the first time they eat a piece of peanut, they get a reaction, an allergic reaction. And by then, they're all going to be allergic. So we don't know what happens before. So my research was to actually study what would happen and why do people get allergic. So I did a lot of work with mice, in other words, and flies. And flies. I studied fruit flies. Fruit flies, the
How
little
would you
teeny ones.
yeah.
Yep,
Who are you
Microscope.
that's right. So it's called doing fly pushing. You have, so flies don't die when you give them carbon dioxide. Like fruit flies, they don't die when you give them carbon dioxide, they just fall asleep. So we would give them carbon dioxide, put them on this pad that had continual carbon dioxide coming out. Then you use
So they would stay asleep?
they'd stay asleep, and then look through a dissecting microscope, and you have a paintbrush, and you literally just push them. So it's called fly pushing. And you separate your males from your females, and you can. I've worked with everything from fruit fly embryos to larvae to pupae to the actual adult flies. Done it all.
And what were you studying flies, like using flies to study?
So when I started, I actually started studying oogenesis, which is development of the eggs in flies, so female flies, and then I studied spermatogenesis in male flies, and then I studied embryogenesis, which is development of the embryo itself. Yeah, itty bitty flies, and I. Fun fact,
So, our
it's-
final question for you for this day is, what is your view of this problem? What's your view of bone density loss as a problem?
What's my view in terms of like whether it's necessary?
No, like what's your view of this problem? Like, do you believe that it is a hard issue to tackle, or do you believe that it is a do you believe that enough is being done about it in the current state?
Do you think it's like relevant?
It's definitely relevant. Definitely relevant. Is there enough being done? I don't think there's enough studying, research, funding being given to fully understand how the biological processes change. When you go into space, there's a push that we want to go to Mars, but there's not enough being done to actually understand how your body changes at the cellular molecular level. So there is, yes, we know that bone density loss is an issue, but we're not spending the time to understand how the stem cells are, how they themselves are changing. I definitely think that's a relevant question. It's an important question, and it's a question that needs a lot more attention. So.
And to add to that, whether you're comparing it to bone density loss in space or even on Earth, we don't have enough data. Like with the astronauts who go, we have only a handful of astronauts, maybe 12 to say 20 or 30, and over the course of several decades of launch. But then even on Earth, humans, as you age, bone density loss is a reality. Osteoporosis happens, and so understanding how
Your immune system
that
changes.
how the body is affected, what does that mean,
Yeah.
is still needs a lot more work. And so on all fronts, bone density loss or understanding it is critical.
Yeah.
Because that helps us prepare.
And a lot of the research for it is currently taking place with mice, which is a good starting point. But I'll also caution you in that whenever we study mice, it's a good starting point, but that just because something happens in mice does not mean it's going to take place in humans. So yes, we can start off studying in mice, but only about 10% of what happens in a mouse is going to be equivalent to what happens in a human. So yes, we can suggest proteins, we can suggest this is how expression levels change, but until we study it in humans or take human cells, it's very difficult to know this is exactly what's going to take place. And I think that's at least 20, 30 years of research.
So my question to you is, what is common between a mice DNA or structure and humans, and what is different? When you say
In
they use
terms
mice.
of our genes, like if you actually look at our genomes, we have about 95% similarity in genes.
Okay.
So that's why mice make good model organisms when it comes to studying. Before we do anything in humans, they are a good start point.
Okay.
What's different is that how their immune system works is slightly different than how humans works. How certain cells and expression is different than what it is in humans. Otherwise, we'd all look like mice, right? Like we need mice to look like mice, and so expression levels of certain genes is different with mice than it is with humans. But if you look evolutionarily, like even as embryos develop, you can see the similarities from like chicken eggs to mice embryos to human embryos. There are stages and they all look the same. When it starts to differ is when you're actually changing into a human and having, instead of us walking on all fours, we walk on tubes. That's how evolutionarily we've changed. So those expression levels have changed. But when it comes to medical research, anything that we do in mice, if a mouse is going to stay healthy and it's going to stay happy, that is our first step before we can go into the clinic to try and put this into a human. And only 10% of those are effective. So there is a huge gap still, but we can't study something in just a dish and then be like, well, we're gonna put this into a human. We have no idea if people will die from that. So that's when we go into mice first.
Okay. So you said that you were a lecturer at McMaster University.
Mhm.
So what do you teach there?
Advanced cell biology. Third year advanced cell biology. I.e.
Your sister took that
Yeah, last
class
last year.
I didn't teach a class.
My PhD supervisor taught it last year though. Yeah, it's possible.
She's in 4th year now, so
Nice. Is she doing
art side.
art sci? Okay.
So that concludes our interview, and we'd like to thank you for taking your time to be with us today and answering our questions.
Informative for you guys.
Yeah.
Cleared up some doubts.
Yep.
Yes.
I learned a lot more on the health side of all of this, because I was like, that is great, but I wasn't aware of the complexities that come into adding more into a DNA versus expressing it. So,
Mhm.
yeah, I learned as well. So thank you.
No problem. My pleasure.